figured out and we actually derived quantitatively a nice expression where we showed that for macrophase separation, we saw that our critical chi-n, so the critical value is equal to 2. For microphase separation, for a 50-50 block copolymer, we saw that chi-n was equal to approximately 10.5. But now, and we kind of understood this, right? Because here we just had this delta S of mixing. That's what drove it to the mixed state, as opposed to the phase separated state for macrophase separation. We know that this term is usually pretty, again, small for polymers, or this contribution, the magnitude here. And for block copolymers, obviously, it would be small as well. So really what causes this increase, this fivefold, more than fivefold increase in our critical chi-n value was not only this delta S of mixing, but the delta S of stretching term. So in order to microphase separate into that lamella, those chains had to be stretched, and they stretched proportionally to n to the 2/3. Very stretched, even compared to a good solvent. So this additional, that stretching term, that's what caused we need to be even at higher chi-n values, lower temperatures, in order to microphase separate. Now, this is kind of a phase diagram for block copolymers, and we're varying just basically the volume fraction of, let's call it our A block here, versus our B block. Now, when I'm at 50-50, I'm at basically this lowest, everything is disordered in here, as we kind of see, everything is disordered around there. But once I cross over this line, just like our binodal and spinodal, you are all polymer phase diagram experts, I begin to, basically, this is a microphase separation diagram. So, right at here, when I'm at 50-50, I could go into this lamellar region. L is lamellar, G is like your gyroids or double gyroids, cylinders, spheres. We've seen this before in our, class lecture notes as well. But I'm really interested in this behavior because look at the chi-n for microphase separation for spheres. It's even greater than that for lamellar. So why does this critical chi-n increase if I shift my basically AB fraction? Well, let's go back and let's consider what were the free energy contributions that led to microphase separation in the first place. So I like this figure here, if I can find that figure. Right here. So we said the push for the disordered state is the stretching term, and the push to be ordered is basically this enthalpic term, so the number of those AB contacts, right? So my critical chi-n, so basically the strength of these interactions, this chi-n-AB. So as my chi-AB, if it's really, really, really large, they really don't want to interact with one another. So what this is saying, and this is also, but remember, this also kind of gets multiplied by the volume fraction of A and B as well. So we'll get back to that in a second. So what's happening here? Why does my critical chi-n have to be so much larger as I shorten? Let's draw a schematic here for my spheres. So what happens here is my chain gets, and then it's long here. So when this phase separates, and again, it's going to kind of form into kind of basically some, sphere, as we can kind of see way back at one of the beginning stages of our, it's going to form basically right here. So what's happening? Why is the critical chi-n for microphase separation so much higher? Well, to microphase separate, we want to maximize the number of AB contacts. We want to minimize those unfavorable AB contacts. But here, we need a larger chi-n driving force because there's fewer AB contacts, right, than in that 50-50. Because again, it's going to kind of, your volume fraction is not the same. So your number of AB interactions, so those are not going to be as probable, because again, when you think about the chain here, it's 50-50 mixture. It could flip around and hit those. So that's one issue that's kind of driving us. So the number of AB interactions might be slightly kind of less. But the bigger driving force here is what? So what leads us to this disordered state? It's the stretching here, right? So here, the polymers are stretched. But when they're in this kind of spherical configuration over here, so the number of AB interactions, so we don't have as many, so there's not a strong enthalpic driving force to phase separate. But here, we're not only stretched, but we are curved as well. So we're inducing like some additional curvature into kind of, again, forming those kind of spheres. So again, there's stretching here, and those number of AB contacts are less. So there's some additional curvature pulling, so it's not only elongated or stretched, but they can be, one can be compressed, they could be, again, inducing curvature is very energetically unfavorable in our system. So that's going to be pushing for that delta S of stretching term is probably going to be larger as well. So we have larger delta S of stretching. We have fewer number of AB contacts potentially. So that's what's going to drive this critical chi-n to increase even further. So I really like kind of this intuition in this phase diagram, so you can kind of see what's happening here. Again, we're not going to derive the entire phase diagram or derive the kind of critical chi-n and those for this whole phase diagram, but this is just, again, it's a general formalism. You can apply it to the same to these different microstructures, these different microphase separation. But again, this is our toolbox, our toolkit. We could change chi A, B. We could change our volume fraction A and B, and we could get, from just these two parameters, we can get these unbelievably different microstructures. And really just from these two parameters here, varying them, phi A, phi B, and you can control, again, with that, you can control that lamellar spacing, or again, whatever periodicity, whatever spacing you want, or the microstructure that you want for your microphase separation. So there are definitely defects, and you can kind of read about this. And again, if people would like these lecture notes, again, you could pause the video or you could read them. But there are defects in self-assembly, but you can reduce those defects with some strategies like applying a bias field, substrate patterning, constraints, so like walls or trenches when you grow your black copolymers. We talked a little bit about graphepitaxy kind of last time in the Carolyn Ross group. So those are some really interesting kind of values. So in the next, not values, very interesting research that you could kind of look into to control microstructure. But next time, we are going to get into black copolymer, homopolymer blends, specifically micelle formations and critical micelle concentrations. So several more derivations next time, but again, very interesting, very qualitative, and very applied derivation. So micelles are very, very important, especially in the context of lipid bilayers for my biological people and my bioengineers. So we'll get into that next time. All right, I'll see you all then. Thanks. Bye.